Ladies and gentlemen, we are live, coming to you from Matt Buchanan Studios, brought to you by me, Matt Buchanan. Follow us on Facebook and YouTube at Matt Buchanan Studios and Matt Buchanan Comedy. Check the episode description for links to all of my, as well as my guests' social media handles. Quaco Inc. is the official sponsor of the show. Quaco is also the official sponsor of my upcoming book release entitled An Explanation for Life, the Universe, the Brain, the Mind, and Consciousness. Available soon from over 30,000 retailers in over 100 countries. Quaco is also the official sponsor of the hot new board game, 33 Degrees of Order and Chaos, coming soon exclusively to Amazon Prime. This is Laugh Therapy, and I am the Comedy Doctor. Stay happy and healthy, folks, and enjoy the episode.
Roger, 0J, and I feel fine.
We're live at Matt Buchanan Studios. Here we are today. It's like the end of March and in quarantine right now. Gonna do a recap episode for my Boston trip, the Harvard interview that I did there, and the Harvard lecture that I did there. And I think I said I was gonna do this episode like weeks ago, kind of taking advantage of the fact that nobody cares, I'm guessing. At least nobody's in a rush for it. So I took my time, basically, and waited to hear the outcome before I did all these things. And so by the end of the episode, you'll know what the outcome was of my Harvard interview. So that's exciting. And what I'm going to talk about today in this episode is basically what went on with the Harvard interview. Like what is it? Why did I get it? What was it for? What was the position about? And the lecture that I gave while I was there, I'm going to give here, and you can see the slides for it right there. And I'm just going to start with a bit of a brief background before I jump into the slides, just to give the story about Boston. So maybe about a month and a half ago, at early February, in early February, I saw an application for a postdoctoral fellowship. That's the official title of what the job is for my academic friends. They'll know what I mean as a postdoctoral fellow. For non-academic people who aren't in that field, basically what it's called, or what a postdoctoral fellowship is like a scientist position. So you'd be working as a full-time scientist, usually for a minimum of 2 years on a postdoctoral fellowship contract. And it's sort of like one of the next most logical steps to becoming a professor. A lot of people will do like 1 or 2 postdocs, which means they complete, 2 or 4 years, sometimes 6 years of like full-time scientist type positions, working in different labs and publishing. And then, so again, you're full-time, but it's, you're not a professor yet, so to speak, or a full professor. So it's like a next step to get you there. So that's what I'm looking for. Now I'm at the first stage where I'm allowed to even apply for that sort of thing. Because I'll be finished my PhD very soon, I'm eligible to apply for those jobs. And so I've been looking for, great opportunities like this one. And it was in a lab at McLean Hospital. That's the psychiatric hospital of Harvard Medical School. And I had an interview over Skype, and I sent in my application. A couple of weeks go by, I got a call for a Skype interview, or email, same thing these days, and we talked for about an hour and a half. It was a very good interview. Everything was quite positive. About a week later, I got a call back to fly to Boston to go and meet the lab and do an in-person interview. Now, the reason I said I wanted to explain like what it means to do the interview is because it's not quite like a traditional job interview when you're applying for major scientist positions. What happens is, so basically it was like a 9-hour interview. It's a full day thing. And people were like, well, how do you get interviewed for 9 hours? And basically it's just because every person, like a whole team of doctors. So by the end of the day, I'd met with like 15 different doctors. They all interview you for about a half hour each, sometimes 1 at a time, sometimes 2 at a time. And it's not like super intense grilling you the whole time, but it's so they can ask you questions about your research. They can learn about what you're trying to work on, what your research goals are, future goals are. And then it's a way for me to learn about them as well. So each of the persons who I met with were, part of the staff who work in the unit, the lab that I would be in. Some of them, several laboratories that make up the whole unit. But ultimately, they're all staff at the end of the day. Some of them are professors, full professors, assistant professors, postdoctoral fellows, and like PhD students, and a team of research assistants that I met with as well. So basically, you meet the whole team. That's what the interview is all about. So that's a full day. You end up meeting everybody. Then I met the big boss man, the man in charge of the whole laboratory, Dr. Pizzagalli. He's runs the center, and all of these people are ultimately his staff at the end of the day. So my job would be to work full time for him. Part of this experience meant that I was going to give a lecture while I was there, and that would also help them evaluate my research. It wasn't just any lecture, it was a lecture strictly based on my research, what I've done for the last 5-6 years or in my research career. So you'll see in some cases, so for me, my research career is 5-6 years. There was likely other candidates presenting and doing lectures who were also interviewed for this that might have had 8 or 10 years experience of research. So the competitive level is very high, of course, and it's quite a honor just to be able to go there and give this lecture. I was very happy about that in general. Already I was excited and looking forward to giving my research and sharing that with like-minded individuals, people who could actually get value from it for their laboratory. So all that being said, if this is before COVID at this point, so travel, everything should be fine, but you probably don't even remember because everything's so overwhelmed since then. But right before this end of February, there was a huge snowstorm in Ottawa, and well, I guess if you're not from Ottawa, then that you wouldn't know about that anyways. But there was a huge snowstorm. So my interview was on a Friday morning in Boston on the 28th of February, and I was supposed to arrive Thursday morning, give myself the day to kind of get prepared, situate, work on my lecture, practice it, and that sort of thing. And 3 flights got canceled, one after the other, all because of the snowstorm. So I was supposed to fly out Thursday morning and then reschedule the afternoon and reschedule it to the evening, just waiting and waiting and waiting and back and forth from the airport and waiting at the airport. And then finally got sent home Thursday night, no flights going out tonight. Okay, so very discouraged. At this point, I was sure I was going to miss the interview. I thought there's no way. I managed to get on the first flight the Friday morning at 7 a.m. Didn't end up leaving till 9 a.m. My interview was supposed to start at 9 a.m. I'm obviously this is not the most ideal situation for me. I should mention that on Wednesday night, I reached out to the staff, the research coordinator who was sort of handling my trip there and sort of taking care of the administrative end of my going to Boston. And I notified her that on the Wednesday evening, before even I got canceled, I saw the first flight got canceled already. By Wednesday night, they canceled the first flight on Thursday. So I was already thinking something, bad could happen. And anyways, so that happened. I did notify her, so they were at least aware that I might be late. So I didn't totally get screwed on that account. They were at least aware. They were very flexible and very nice and accommodating and helpful to the fact that I was there 2 hours late for this interview. But I made it out. I got there Friday morning, 7 a.m. flight. Didn't leave until 9 a.m. Ended up getting there by like 9:30. Changed in the airport bathroom to get into my suit. Got into an Uber, which was like probably $100 US Uber, just to get there as fast as I could. Took me straight from the airport, got me to the McLean Hospital, and I arrived. And this is where the story begins, and I got to meet everybody, and it was a really wonderful day. And now I'll give the lecture that I gave there, and basically you'll learn a little bit. I'm going to not give it exactly like I gave it there. I will keep it sort of medium level, so for my academic friends who are watching it. They can get that value out of it, but also make it explanatory enough. I'm not going to go into all of the nitty details. It was an hour-long lecture, and I'm not going to spend that long on it. But it's just to give you an overview. This is my research. This is what I've done for the last 5 years or so, and I know I talked about some of it to a little extent in the last episode. Well, not necessarily the last one, but in episode 1, my first, the solo episode that I did. So here, I'll present what I did there. So the name of my talk officially was Discovering New Electrophysiological Biomarkers for Enhanced Endophenic Classification and Non-Invasive Brain Stimulation. I'll break that down real quick before we even dive into it. So what that means is, what I'm trying to do is I'm looking for new biomarkers. So a lot of people might be more familiar with the idea of a blood marker. So right now, okay, COVID is a huge thing. There might be, you take a blood test, you can look for something in the blood that indicates that there is a abnormality, or you can specifically find a virus or a certain disease. It works. You might have, if you've had like a sexual health test and you've done an STI test, like, you give blood and or urine in some cases, they can find a marker in that. There's stuff in there. So that's the same concept. This is an electrophysiological biomarker, though, which means it's a signal based on the electricity that comes from your brain that might be unique according to specific populations. So you might have a unique physiological signal if you have depression. You might have a unique physiological signal if you have schizophrenia, or if you have post-traumatic stress disorder. ADHD is the one I'll be talking about to an extent today. And we can measure that signal. So by finding that signal, you can diagnose people better. You can create better treatment options, more targeted treatment options, because it's specifically more tailored to their individual proclivity. Now, that's the idea of the enhanced endophenic typic classification. So phenotypes are like subtypes of diseases. So I did mention this in one of the previous episodes. So if you have depression, not everybody has the same type of depression. There's subtypes of that. Another scientific way of describing a subtype, or basically a synonym for this, is phenotype. It's like a type of subtype. And an endophenotype is like another layer on top of that. So here we're looking at these very specific phenotypes, and we're using these biomarkers to identify them. And then what brings it all together there at the end is the non-invasive brain stimulation. So the brain stimulation that I do, it's like a very low mile current, uses up to about 2 milliamps, which is like thousands of times less than even what you get out of a cell phone. So it's a very low current to alter brain activity, and you can apply it just on the scalp. And it's non-invasive, which means, that's what I mean by on the scalp, just on top of your head. You don't have to do surgery. There's no open wounding or nothing like this. It's just put these 2 small electrodes like this big, you put them on the scalp, and it can help alter brain activity. So now we want to pair all that together. We want to take the brain stimulation and then use these endophenotypes and these subtypes and the biomarkers, the electrical biomarkers, pair that with electrical brain stimulation, and then put that together so that we can specifically alter or correct the abnormal electrical activity that is going on in the brain that we've identified. So that's what I've done in short. That's the whole story. Now, see, this is it. This is the photo the day that I was supposed to fly out. I took this photo. This is what my backyard looked like. And I made a joke that it'll be nice to go skiing, and I did. I went skiing a couple days when I got back, or no, this was, I think, taken before that, but I did go skiing once I got back, once before everything went to shit. So before I describe exactly what my research is, I usually like to preface that with a background on the present state of the research field and what led me there, because I didn't just come up with all this on my own. I mean, it's based on theories that other people have had and research that other people have done. So you can see this paper was published January 2016. This is a paper from the laboratory that I was interviewing for. Did I put it in there because it was relevant? Absolutely. Did I put it in there because it was a paper that I was from the lab I was interviewing for? Also, yes. Both of those things, I think, make it even more relevant. So, also, cheers to this episode, because I can have beer, which, I mean, I don't suppose that would have went over well during the live lecture, but So this paper, I'll just read the title: Neural Correlates of 3 Promising Endophenotypes of Depression, Evidence from the EMBARK Study. And I'm looking over here because I have a prompter over here, but I also have some notes over here. So that's that. And so the EMBARK study was like this big study that was done on depression, from the laboratory that I was interviewing for. And what they found was neural correlates. So same idea as what I'm saying, electroneurophysiological biomarker. Neural correlate is another way of saying that, but it just depends on what neuroimaging modality you're using would determine sort of how you call it. So we're using electroencephalography, and therefore it's called an electroneurophysiological biomarker. If they were using something else, in this case, they did use EEG actually, but they also had some fMRI, and so you might call it some other way. It depends on also the scale. So here they mention what they found was that in the anterior cingulate cortex, theta activity. So theta is a type of electrical activity. Basically, it's a frequency of electroactivity from from 4 to 7 hertz. So it's when you measure your brain activity and the electricity of it, anything happening in the 4 to 7 hertz range is called the theta activity in the brain. And they found that theta activity in the anterior cingulate cortex could predict depression. So they found, and they had these endophenotypes. So like we were talking about these subtypes of depression, they found 3 endophenotypes that could be described on major depression by looking at different brain regions and different frequencies. And that was a great discovery, which kind of inspired some of the work that I did and the approaches that I was taking to get this sort of cutting edge approach to clinical neuroscience and this translational neuroscience. And here you see another paper from the same group, or sorry, not this. This isn't the same group. This is from Andrew Drysdale and Connor Liston, who I definitely talked about in a previous episode as well. And this is their paper, Resting State Connectivity Biomarkers Define Neurophysiological Subtypes of Depression. Same thing we just talked about. They found 4 subtypes of depression, this time using functional magnetic resonance imaging, otherwise commonly known as fMRI. And they had over 1,000 subjects, 1,188 you can see, and they classified diagnostic biomarkers with up to 82 to 93% specificity and accuracy. And this was done at multiple sites and in different laboratories and different hospitals. So very impressive, exciting paper. This was published in Nature Medicine, I believe. And then Connor Liston did the follow-up study of that with TMS, transcranial magnetic stimulation, and showed that he could treat people better based on their subtypes that they discovered in this study, in this new one, and then they published that in The Lancet, 2 of the best journals. So I'm just saying that quickly because I talked about that more at length in episode 1 when I was kind of describing clinical and translational neuroscience. So part 1 of my research. It might be a little bit hard to read, but I hope that it's clear enough, and I'll read it out. So study 1, I did a systematic review of the safety, tolerability, and acceptability of TDCS. That's transcranial direct current stimulation. It's a type of electrical stimulation, very mild current. And I did a systematic review of the safety, tolerability, and acceptability of this in youth. Then part 2 of the study, we did an RCT. That's a randomized controlled trial. So you might have heard this term before in the news or when they're doing new drug trials. You do these randomized controlled trials. And ours was also a double-blinded trial. So there's different layers of control. So basically, we add control by randomizing things so that there's no effect of, any a priori effect. We randomize the amperage that the kids received. We'll get into that. And that was based on the side effects that children would experience. So the reason we did this whole study is because in all of these studies really at the beginning was that there's so much research on brain stimulation in adults. If you look at all the research that's been published, like thousands and thousands, tens of thousands of papers and studies have been published, only 5% of any of that research has actually ever been done in pediatric populations. And so my sponsors, I'm from the Children's Hospital here in Ottawa, and so we had certain motivations and hypotheses that were about using brain stimulation in children. And this was early 2012. I think some of the ideas started with my PI, the principal investigator, and then I took it up in 2014. and completed these 3 studies as well as multiple other studies along the way. So everything I'm presenting today, I should know, is not actually everything that all my research. It's just, to date, some of my most impressive studies, you could say, some of the better stuff that I've had the opportunity to work on, although I found it all quite exciting. So we did these studies because there was so little research in children. So we had some hypotheses about doing studies and using brain stimulation to help children and people with mental health and neurological conditions, not just in adults, but doing that in children. But when you want to do a study, you have to submit to a research ethics board. And this is a good thing because it means that they make sure everything that you want to do is ethical and under strict guidelines. We did that, and we submitted an idea that we were, to try to use TDCS, as a way to treat people, children with mental health and neurological conditions. First, they came back and they were like, well, a bit hesitant logically and fairly so. And, there's some back and forth. And then the discussion led to, well, perhaps we should define first the safety of TDCS in children in these populations. Now, when we started this in 2014, there was barely anything on this, like hardly any studies published. Now in 2020, there is a good handful, maybe 20 studies, but 20 is a very low number compared to the hundreds or thousands of that you would expect in this case, to really have that be this technique become commonly used. So we started off by doing this systematic review, which meant we reviewed all the literature. So we went to databases like PubMed and databases like IEEX and Google Scholar and Scholar Portal. And we looked with keyword searches, things like TDCS, safety and tolerability in children. The same thing, but in adolescents. And then we would do the same variations of that. Like we'd spell out the words transcranial direct current stimulation, tolerability in children, in adolescents, and so forth. You use all these keyword searches, very systematic. You have a protocol, you follow it exactly. You do the exact same search in all the different databases. You get hundreds of results. You break those down and you apply inclusion and exclusion criteria, and then you get a final result that really tells you a lot about the current state of the literature. So that's how it all started with us. When we did this randomized controlled trial where we had 30 children who each did 2 sessions and 30 adults that each did 2 sessions. I'll explain the logic of that later. And then we followed that up with a third study where we actually interviewed all the families of the children. So half of our child sample had ADHD or another mental health and neurological condition like Asperger's or autism, anxiety, and so forth, sensory disorders. Then we interviewed all those families to discuss their experience with TDCS. So this is a qualitative type study. So we wanted to understand firsthand what the parents' experience was of TDCS in their child. What was it like for them? Is it something that they could see? Because none of them had ever been exposed to it before. Nobody had experience with it. It's a very new technique. So they're all used to things like medication. They would take or have tried different medications. And they were very motivated and encouraged by the fact that there is perhaps an alternative treatment that could work for them that isn't medication and that might actually have a positive effect in treating the child. So that was the third study that we did. Part 2, I'm going to talk about some of the EEG studies. This is where the biomarkers come in. So the first thing was, here's all the brain stimulation stuff and looking at the safety of all that in kids. And then here's how we can try to connect some of that together and look at some other research that I did with EEG, the electroencephalography, which is a type of brain scanning where you put these electrodes all over the head, usually anywhere from 32 electrodes up to 256 electrodes. In this study, well, actually, in the first 2 studies, we actually used 19 electrodes, so you can use less. And then in the last studies, it was 64 electrodes. So it's a fair range depending on what you're trying to do. In clinical practice of EEG, not in a neurology epilepsy clinic. In an epilepsy clinic, you would use a full grade of electrodes, but in a QEEG type practice, like the private clinic I work in for brain injury, 19 electrodes is the common. And so we've done a lot of research on that. So 2 of the things I'm presenting here actually aren't even part of my PhD. Like they're not even in my dissertation. They're projects that I did, research projects that I did during my tenure as the director of research at a private medical clinic here in Ottawa. and I still am collaborating on that because everything, basically all the papers I'm talking about today, some of them are published already in scientific journals, and the rest of them are, some of them are in review right now, waiting to be published, like in review with the journals. Like we've already, it's been submitted, and some of them that are being submitted like this spring very soon. So all very exciting time to get this stuff out and see what comes next. So in study 4, it was again the using electroneurophysiological biomarkers for non-invasive brain stimulation, what we talked about. Study 5 was a epidemiological study where we made a discovery at the clinic, the first account ever of something called the polytrauma clinical triad in civilians. It's been commonly reported in post-combat military, but this was the first account of it ever being reported in civilians. And so that was an interesting. That paper has already been published in the Journal of Pain Research in 2018, and then we kind of, it's not a direct follow up, but it's the same clinic, same group that I was working with, and we did another study where we had EEG, electroencephalography, in 57 patients and compared that to 57 controls. So all this is, I'm going to explain all this more. So I just wanted to give a brief overview of what I'm doing and what I'll be talking about today. So all the brain stimulation stuff that I'm talking about, like I said, some of it's published, some of it's in review being published. In 2019, about 1 year ago, 25th of February, you can see, I hosted, so I was the chair of an international symposium on brain stimulation, and that the international symposium on the advancements in pediatric brain stimulation. I think that's, neurodevelopmental brain stimulation. And we had speakers from all across the world. I was a chair with Paul Croker from the Mayo Clinic in New York. We had speakers from Oxford, from Australia, from New York, Canada. Where else here? Japan, and from Alberta. Some of the top, basically all the world's top researchers in pediatric brain stimulation. Like I said, only 5% of the research. When we did that systematic review, our final thing ended, like our database ended up with maybe, I think there was less than 20 papers in there of studies about kids and evaluating safety in brain stimulation. Like 20 studies. Like to the public, I suppose that might seem like quite a bit, and it actually it's substantial evidence. But 20 studies compared, like if you look up the safety of methylphenidate, like the common drug to treat ADHD or something, or you look up the common drug to treat depression, even though brain stimulation can also be used to treat depression, like there's thousands, like the amount of research is, it's just very scarce. So there's not a lot of people in the whole world doing pediatric brain stimulation, and we had the top of the top here to do this symposium, and it was held in Canada this year. It's supposed to be in Singapore in 2020, 2021, but well, I guess that might fly. Hopefully, things will be back to normal in 2 years. But so these papers have, as a trio, the 3 of them are being published separately, but they've also been published together. The 3 abstracts and the talk has been published in the Journal of Brain Stimulation, with courtesy of my team and everybody there, my professors and the research assistants that worked with me, and we made the whole title here. It was, this is the title for the journal, it was Making Transcranial Direct Current Stimulation in Atypical Child and Adolescent Neurodevelopment a Reality, Translating Safety, Tolerability, and Acceptability Evidence from the Laboratory into the Doctor's Office, the Classroom, and home. It's so way too long. It's the longest title probably ever published. I don't think there's a longer title, but first of all, it's definitely explanatory. It's clear. I mean, maybe not the first time I just read it out, but it tells you what you're trying to say. But that was the point. I was like, okay, well, we're publishing all 3 papers into one. We got to make a title that's reflective of that. But also, if they're going to research, or if somebody's going to come across it, well, if they don't read the whole thing, if they at least read the title, they basically know what the whole thing's about. And then it was published again. So I presented this stuff at the Ottawa Royal Mental Health Institute and the Young Researchers Conference. And in 2019, I won the award for Best Clinical Research. And then it was published in the Journal of Psychiatry and Neuroscience, the same 3 papers. And actually, the next studies that I was talking about too, with the private clinic in 2017, I also won Best Clinical Research from the Ottawa Royal Mental Health Institute for those studies. So I've had good luck there. It's been a great opportunity for any young researchers to present what they've been doing. It's an absolutely fantastic venue. Unfortunately, the conference this year was canceled, but maybe it'll be rescheduled. I don't know. It's a lot of work that they put into it, and every year's been great. Like Robert Sapolsky came and spoke at one. It was incredible. We've had Jonathan Downer and Paul. I was talking about and Connor Liston came and gave their talk, which was this sort of duo brain stimulation, fMRI, phenotype, treatment talk. Absolutely incredible. And yeah, it's just been great. So I've been lucky to go there. And last year I was like a mentor there as well. So it was like a speed dating thing for young students, which sounds bad. I wasn't dating the young students. I was like a mentor where they came around and had a chance to talk to different mentors. Like a group of 3 students would come to each table with one mentor at each table, and we did that for like an hour or 2 or something. And I'm just kind of telling them about my path, my journey, what I've been doing, and what I work on, and that sort of thing. So this is me. This is the brain stimulation. Like I talked about it, you can see me getting the stimulation. It's just these 2 little electrodes. That's in the laboratory, the neuropsychiatric lab at the Children's Hospital. It's non-invasive, 0 to 2 milliamps. That's it. What it does, it pulls negative ions one way and positive ions the other way, and it makes the likelihood of an action potential increase or decrease. So under the blue electrode, you're getting a decrease in activity. Under the red electrode, you're getting an increase in activity. And you can pull that one way or the other, and you can position the electrodes necessarily toward whatever brain region, anatomically based on just the scalp, requires manipulation. So you might hear about a lot of brain disorders like, oh, this brain area is not active enough, this brain area is too active. We can alter that with this technique. It's about achieving this interhemispheric balance, and by just providing light stimulation, we can do that. So I mentioned this already. This was the TDCS systematic review on the safety, tolerability, and acceptability in children. Our methodology was largely based on a previous paper that was published on the overall safety of transcranial direct current stimulation, not focused on children. They did review child literature, but it wasn't focused specifically on children. This was published in 2016 in the Journal of Brain Stimulation by Meron Bixum and many other researchers who contributed to that. But for Meron Bixum's group from New York University, and we actually plan to contribute so he has a site that you can go to called tdcsdatabase.com, which basically holds all this data. It's open source. You can go and use it and write your own papers based on it. So if you look at it though, there's like more than 500 papers in there reviewed, and with all this data out and extracted, absolutely fantastic resource. But if you look, the only thing that is really missing, there's a lot of empty in all the child sections. So they'll have the name of the paper, but none of the data is there. So we're looking forward to being able to provide that through our study, and that'll be coming out soon. So yeah, we looked at these 4 databases. The main thing that we wanted to evaluate, we looked at hundreds of papers, and what we wanted to find was studies that were done in children under the age of 18, studies that were specifically designed to examine safety, tolerability in children, and studies that reported on side effects and used neuroimaging. So we wanted them to actually have an intent of evaluating side effects, and not just like a tertiary outcome that they happened to, they were investigating depression, but then they looked at side effects on the side. That could be appropriate as long as they had a part of the study that was built to measure side effects. Here's the flow chart that we, based on all the stuff that we reviewed. So we started off with 440 papers. All of those were reviewed, and then we excluded so you review all the titles, the abstracts, and the keywords. Then you eliminate all the papers based on that doesn't fit your criteria, basically. And then that led to 78 papers left over, and then eventually only 22 total in the full text review, and then 8 that are actually being published in the manuscript. and another 13 that are being published as a data report. Well, pardon me, not published, so they're being published together with everything as a data report, which is going to represent the database that we're contributing to the overall the TDCS database from Aaron Bixum. Oh, here's all the so when you're doing a systematic review, if you've read a scientific article, or imagine any article that you've read, whether it's scientific or not, or it's just a book, anything that you've read that you think might be really interesting, and you want to know everything about that topic for that. And there's so many different papers. So here we end up reviewing, say a dozen papers, and we're going to write about all those papers in one. So we're going to write one paper about a dozen of them, and we're going to kind of synthesize all the information that was there. All these papers did something similar, and we're going to aggregate it. That's what you're doing when you're doing a systematic review, essentially. So you pull the exact same data from all the papers. Every paper reports this data, in their method section, in their results section, and so forth. So the job in the systematic review is to do a data extraction. So you go into one paper, you take all the data points out of it that are relevant to your study. You do that to the next paper, the next paper, the next paper. You aggregate all that information, and now you know something more compounding. You have much more confidence in the evidence because you have evidence from all these papers put together, and it's a lot stronger than having just one of these papers. So when you're really looking for information on a new topic in science or a new treatment or something, you want to know about systematic reviews. You can look at one treatment here or there. It might be promising. It might even be a holy grail, but what you need to see is things like systematic reviews and meta-analyses, which is just like a different mathematical version of a systematic review where you can do additional math on top of it and so forth. So we took out all kinds of data. We wanted to know what tests were used, questions were used, what images were used, imaging, like neuroimaging was used, what cognitive tasks were they doing during the study, what was the method of examining side effects, what was the level of voltage in the brain stimulation machine. Like I said, it can usually range from 0 to 2 milliamps. So what was that? What was where did they put the electrodes on the head? How big were the electrodes, the current density, and what outcomes did they evaluate? How did they measure safety? How did they measure tolerability? How did they measure the acceptability of the technique? So here's just a table. There's no point really going through this. This is a table that we've aggregated for our manuscript that shows a review of all these different studies. So on the y-axis, you can see a number of different studies that have been completed and the years that they were completed and the author, the lead author on that. And then you can see, for instance, the number of subjects that they had. So how many kids were actually in their study, what was the stimulation, and you can see the population is like, what was their condition? What did they have mental health or neurological wise? Was it a randomized controlled trial, yes or no? And it would be boring to go through every little detail, but that's the type of data we went through. This also another boring table to just present, but I'm just showing it for a second to give you an idea of the type of data. So this is another table we created, which is the task that they perform, the mode of side effect examination, and the adverse events and attrition, the acceptability. So how did people leave the study or drop out? Like, did they have such a bad experience that they had to stop the study, that kind of thing? And the depth of data. So what you see on this one slide is for one paper of all the stuff that were reviewed, one table of one of those papers, and we have 4 tables reviewing each of all those. So there was a really significant amount of data. Here's just a cool image of us and some of the kids participating in the randomized control trial. So that was study number 1. Remember, there's like 6 studies that I'm going to touch on overall today briefly. That was 1. This was number 2. We did the review. We found out, according to the literature, of all the papers that we reviewed, there was more than 20 in our database. Everything points to TDCS being safe in kids. There's no reason to believe that it should be not safe in kids, and therefore, we should be able to use it and start seeing it used more commonly. And then we moved on. We did a randomized control trial, and we had 30 kids and 30 adults. and they all participated in our study, and the goal here was to compare the kids and the adults. So remember earlier when I said 95% of the research has been done in adults, only 5% in kids. So in order to create a translational paradigm that would facilitate new treatment options with TDCS in kids, we could base that off everything we know in adults. So we already have all this information in adults. We should be able to translate that to kids. So in order to do that, we made this study where we directly compared the safety and tolerability of a child group, 30 kids with 30 adults. And then we were able to see, are those things different? Are their responses different? Do they have more side effects than adults do or less? And then we were able to compare males versus females and control group, so somebody who is totally healthy versus somebody who might have a mental health or neurological condition. And the easiest way to put it, the results, and the quickest way is that no, there was no differences. Not out of 234 comparisons, there was, I'm hoping I'm getting these numbers exactly right out of my memory, there was 11 that showed to be any different at all. And that was things like it felt slightly more itchy for a child, but the week before in their baseline was more prone to crying, for instance. So not even anything to do with the stimulation. Like a week before they had stimulation, if you compared their response, because we did follow up like a longitudinal, we recorded data from the kids at a week before all the way till a week after, as well as before and after every session. So we found some differences, which said, for instance, well, 1 week before, children were more prone to crying than adults were. That had nothing to do with the stimulation. So all this is to say, out of 11 or 234 comparisons, only 11 were actually different, and some of them were like that. That's less than 5% of the overall comparisons. And in a statistical margin, 5% is considered the margin of error, which means that there's literally no more than even a random amount of side effects different. So why is all this important? It's important that we did this study and we showed all these comparisons. So I'll just jump into the graphs here to show that there was no difference between the kids and the adults, because then we don't have to redo every study that was done in adults and kids just to see it happening in kids. Now we can be confident that, oh, if they respond in the same way and they tolerate it in the same way and it's equally as safe in adults as it is in kids, well, then we can start to adopt what we have already done in adults and just start doing that in children now. So it's a real translational paradigm to hopefully be efficient, save time, and be cost effective. So this graph is comparing all 6 time points. So we saw them again at the week before, the day before, immediately before and after every time they had stimulation, a week later, and they had 2 sessions. And so we followed everything. We did the same thing for the kids and for the adults, and they had 13 different side effects, and they were rated on a scale from 0 to 5. And the most important thing to note above all that is that on a scale from 0 to 5, where 0 is nothing, 1 is very mild, 2 is mild, 3 is moderate, 4 is severe, and 5 is very severe, on average, every side effect for all of these time points in every case was below 2, below 2, which is mild. So even though there might be a very scarce difference between a child and an adult here, that's still like splitting hairs between saying, well, for them it was mild and for them it was like very mild. You know what I mean? So all things here even point to it being very safe overall. That's the main message to take home here. Like I said, the only thing different about these graphs, this graph is pediatric versus adults. This graph is females versus males. So it's showing the same things, females versus males comparison on the, and this graph showing healthy versus clinical population and how they experienced it. So another 2 things that were significantly different of those 11 that I already talked about, some of them which were irrelevant because they were in the weeks before, and the 2 that were, just offhand, that were in this one, if you look at trouble sleeping and if you look at concentration, the concentration in green for the clinical and trouble sleeping in orangey brown at the far side, the last one for the clinical, you can see that they had more. So concentration in a group of patients with ADHD, we found that was different. So even of all the things that, we're not finding anything different, it's definitely, well, encouraging to say, so to speak, that what we did find different is exactly what you would expect. The only thing that was, okay, the level of concentration of children with ADHD was different than the healthy group. If we didn't find that, that would be abnormal. It would go against what you would expect. So it's all in line with the expectations. So this is great. Moving on to study number 3 now. So study number 3 was really fun. It was fun because we actually got to meet with the families again, and we took time. Well, we met with them. Some of them we did audio interviews over a teleconferencing system, and we got to really discuss their experience at length, qualitatively, open-ended. We had questions prepared, but it was an open-ended conversation. There was so actually, for some of you who didn't know, that I've probably a lot of you don't know that I did this study, and like that was some of my first exposure to direct interviewing, like interviewing people. So I do have some professional experience actually doing interviews and recording interviews before I started podcasting, but that's just an aside. So we ended up interviewing 8 families, or myself. I interviewed them myself. Those 8 families represented 14 children. So what I mean by that is, like, 8 of the families, they might have had 2 kids participate in the study. Some of them had 2 or more maybe participate in our randomized control trial. So they participated in study 2. Then they agreed to participate in the follow-up study, study number 3 here, where the first one, reminder, was the paper. That was a review. So that was just a review of literature, no live participants. Second study, all live participants, 30 kids, 30 adults. Third study, we interview all the kids who had ADHD and all their parents, well, the mothers in this case, and we find out what their experience was with brain stimulation. And we recorded that, and then those interviews were transcribed manually, and we ended up with, like, I don't remember how many, like a couple hundred pages of transcriptions, and then all those got coded by 2 separate researchers. And what they were looking for was themes, common themes that came up across the different interviews from one parent to the next. They were coded using 3 techniques called open, axial, and selective coding. I'll leave it at that. You look for bits that are very specific, that are reoccurring from one interview to the next, and you aggregate those and you code them, and then you collate all that information and you write a big, nice paper about it. And that paper is in review right now. Okay. So this is the 7 themes that came out of this. So 7 major themes that emerged from all of these interviews that was consistent across them, and that seemed to come up for all the parents, were things like TDCS as a source of hope for parents. So they were very hopeful about the prospect of a new treatment. So these are the 7 themes, and so the source of hope for parents. In other words, they were hopeful that they would they were hopeful that there was this new treatment that could help their child, and that might be something, because they've tried medications, and it might be something that works better than medication, and they're really hoping that this could be something good for them. What is the burden of the treatment, though? So the thing about brain stimulation is that if you wanted it's to receive it as a treatment, currently, like, you can't just go to the hospital to get it. That's depending on the type of stimulation. You can't get TDCS just actively at a general clinic or something like that. It's a very research oriented at this point. But if you were to get it as a treatment, it's something you would have to do, like 5 days a week for 4 weeks. So like 20 days for a whole month, basically. But then in theory, this is kind of what the research is pointing to at this point, is you can maybe do that, but then you don't have to, do it for the next 11 months or something. So this was an evaluation of the burden of the treatment. You might say that that's a better trade-off than taking medication every day. Maybe you say taking medication is the better trade-off. We don't know. That's why we did this study. Like, how do they feel about it? That's what we wanted to know. Ultimately, they did. The parents said that they were very encouraging of using TDCS, and they thought that it sounded like a great idea, and that it would be, assuming it was effective. So we'll look at another theme, and we see the bottom theme there: established TDCS efficacy versus established medication effectiveness. So it's efficacy for TDCS because we don't know that its effectiveness yet. Medication, we already know its effectiveness, but it's just the case that some people are treatment resistant and they don't respond to medication because of their underlying physiology. It's nothing that they can do about it. That's just how their biology is, and so that's a main target for a lot of brain stimulation protocols and these alternative protocols, is because people have tried, most of everybody in our study had tried multiple medications and their child still doesn't respond. Some of them do well with medication, but they were still willing to do TDCS if it was going to be better than medication or if it had less side effects. So ultimately, every parent agreed that the side effects of TDCS were barely existent and that the side effects of medication on their child were very intolerable and very undesirable, and that if they could use something like TDCS that doesn't have all these medication side effects, that they would much prefer to do that. So that was an overall outcome of our study. But the factors that weighed into that were also things like the cost and coverage. So a lot of them get medication covered by insurance. So that might not be the case for this, or at least not right away. How do we deal with that from a community and a research and an ethical point of view, especially for children who might need help, who are suffering under side effects, but it's just this new technique that's not covered yet, even though they'll cover medication. So these are things we talked about. And of all the stuff I am talking about, once it's published and it's out there, you can totally go and read it and, and enjoy if you're really into it. And also, an important theme was the education and trust with the care providers. So, what was the perceived compliance of the patients? I'm totally just jumped to another theme. Education and trust with care providers meant that they felt safe with me. Like, I did all those sessions. So we had 120 sessions. We did 60 sessions with the 30 kids and 60 sessions with adults, all conducted by me. And what they're saying is, okay, they felt safe with me. They also felt safe with the fact that I was a representative of a children's hospital and of the lab. Like, if I was just a random student, maybe they wouldn't have felt as safe. But I'm doing a project that's certified with a research ethics board at the children's hospital. This is, it goes a long way, and that these factors helped weigh into their decision about allowing their child to participate in a brain stimulation study involving the safety and tolerability. It sounds a bit potentially frightening, but when you really break it down and you look at the evidence, and when you have an expert's point of view on the field, you realize how safe it is. Now we have the parents' point of view, and they're telling us it's safe and it's tolerable, and they would rather do this in their kids if they could do it in their kids. It's not out there yet. The technique is not clinically available, and for some good reason still. I'm not saying anyone should be premature and do it without the evidence, but a lot of evidence is there, and at-home use is becoming popular. So I mentioned earlier, cost and coverage, burden of treatment. If you had to do 20 sessions across a month, it might be a lot, but there's so many new protocols, even in the last 2 years, from Nietzsche's group, I believe from Germany. I think they're a Germany group, and what they've shown, they've done all these at-home protocols. So they've written scientific articles and done studies where they would do a treatment, but instead of coming into the clinic for 20 days, they would just do it at home, and they would give them a device that they could activate remotely through Wi-Fi or something, and then control it, or XYZ. There's different ways of doing that, and lots of these studies are coming out. I think that's the future for brain simulation in that way, for sure. It's more effective based on the conversation we had with the parents. And then you have perceived compliance. So they thought that their kids would be more willing to do TDCS than take their medication. A lot of kids are reluctant to take medication, especially at least in the sample that we had. And so that was another interesting finding. I'm not going to read all these, but here's just some quotes, because there's no better way for me to really talk about what the families had to say than to just read their quotes. And my favorite quote from all of the hundreds of pages of transcripts was this: "There was nothing about the procedure itself that felt unsafe. I can safely say that there have been no negative side effects for myself or for my children. They've had worse experiences at the dentist." I feel bad for dentists, because I know they already have a high suicide rate, and I don't mean to add to their plate, but it's like, It's true. I've had worse experiences at the dentist. The stimulation is so mild; it goes on your head. You barely feel it. Most people don't even feel it. Some people do. In our study, a lot of people did feel it because we're asking them to feel it. Basically, we're like saying, really focus on what sensations. Is it painful? Is it tingling? Are you getting a headache? Is your vision changing? Like all these things that we evaluated to find out what the side effects are, and virtually no side effects. And the parents felt the same way. Um, I'll read another one here. They said, uh, there, it's difficult falling asleep at a decent time. Both of my children have a tendency to tell me that they don't want to sleep, so it takes time for them to actually fall asleep. Okay, I don't know why that's not the best one to read, but they're because they're on medication, so because they're taking the medication, it's a side effect that they're very wakeful, and so they can't fall asleep. That's what she's saying. Uh, Here's another parent talking about the side effects of and the effectiveness. I like this one. So they say, well, if you compare the medication, for instance, if I put the medication at 100% effective and I put TDCS at 50% effective, maybe I'll say it's not good enough, right? So she wouldn't switch off medication if TDCS was only half as good. But then she says, but maybe if it's 75% or 80% as good as the medication, and because it has so much less side effects, then she would switch over. So these are important things, and this is the type of research question that does still need to be answered. We can't just say, let's just start doing this in everybody. So, that it's fair. This still has to be done. Ultimately, we ended up showing that our results could kind of match up with something called the theoretical framework of acceptability. This is a paper that was published, a systematic review paper. So we've talked about what that is now. This was a systematic review paper that was published about the acceptability of new techniques. So this whole study number 3 is really about how acceptable is TDCS. So we looked at the safety, the tolerability, now the acceptability. And by that, we mean, how do people feel about it? And there's this whole study that was done to evaluate acceptability acceptability of new treatments. TDCS, in this case, being one of these new treatments, and our study that we've published here, and they reviewed all these other studies of different treatments and how they evaluated acceptability, and they found that there was 7 things that seemed to come up regularly, 7 themes that were used to evaluate acceptability, and we kind of matched up with all of them in a way. So they found that there was an effective attitude, ethicality, the burden, the intervention coherence, the opportunity costs, the perceived effectiveness, and the self-efficacy, which is like the compliance. So we kind of matched up with all these in a way, and this was another like incidental finding, but it's good support, just showing that the way we evaluated acceptability was reasonable, and it is supported by previous science and literature. So the take-home messages here: TDCS, it's safe and tolerable to use in kids. That's the one-line message. TDCS is safe, tolerable, and acceptable to use in children and adolescents. And families of children with abnormal neurodevelopment are supportive of the use of TDCS in their kids. And the side effects are favorable compared to medication, according to families who have children with ADHD and who have participated and exposed their child to 2 sessions of TDCS. They agreed, and not agreed, like we didn't ask them that even. They told us that the side effects were favorable compared to medication. So finally, the result of all this, really, for researchers and clinicians, you should be more confident that you could start to do these types of studies, like use this in your clinic.